Hello, I'm Pushpangna, a PMRF scholar from IIT Kanpur. My work is on SARS-CoV-2 to elucidate the SARS-CoV-2 spike protein triggering mechanism for membrane fusion. Owing to the pandemic, we all have a very fair idea of the SARS-CoV-2 virion and its genome. I would just like to emphasize upon the fact that the SARS-CoV-2 genome encodes many proteins which orchestrate its infection and fusion cycle. The SARS-CoV-2 envelope is adorned with spike glycoprotein trimers. If we look at the structure of a single spike glycoprotein trimer, we can see that it has a receptor binding domain which can bind to its receptor AC2 and a fusion stock which mediates membrane fusion. Now, even when we have a plethora of literature on SARS-CoV-2 infection and its infection cycle, there are a few questions which still need to be understood. The first is, what are the triggers for entry and fusion of SARS-CoV-2? And how does membrane fusion take place? And is it dependent on a specific phospholipid? If it is indeed dependent on a specific phospholipid, then single molecule investigation of spike phospholipid interaction during SARS-CoV-2 fusion to target membrane. Now, the key finding from my project, key finding number 1 is that dynamic calcium sensitivity stimulates the evolved SARS-CoV-2 spike strain mediated membrane fusion for enhanced entry. Now, the results of this key finding I have already presented in detail in my previous reviews. The first conclusion is low pH and calcium triggers SARS-CoV-2 spike mediated membrane fusion, and the second conclusion is that evolved spike variants show correlation in calcium sensitivity and fusion activity. The results of this project have already been published in the journal Cell Reports. Now, if I talk about the physiological relevance of the results from my key finding 1, then what happens is that when the SARS-CoV-2 virion is entering inside a host cell, then it primarily follows the endocytic pathway, because what we have found is that low pH or the endolysosomal pH and calcium concentration are optimal for membrane fusion of SARS-CoV-2 spike. This brings me to my second finding, the result of which I have shown in my previous review, which is SARS-CoV-2 spike fusion peptide interaction with phosphatidylserine lipid triggers membrane fusion for entry. If we look here, we have the data in presence of phosphatidylserine and in absence of phosphatidylserine, and we can very clearly see for all these 4 strains that when phosphatidylserine is present, it is only and only then that membrane fusion is taking place. If we look at this infectivity data, which has been performed using the beta-lactamase assay, then we can see that if phosphatidylserine is added externally to the cells, the target cells, which are Vero TMPRSS2, then infectivity of SARS-CoV-2 virions increases. Another important evidence is from this confocal assay, which is to find out the entry of virions inside cell. We have MRFP LAC-C2 in the target cells. The LAC-C2 binds to phosphatidylserine in the membrane, and thus phosphatidylserine will not be available for virions to bind. Now, as we can see here from the white arrows, the places only where LAC-C2 is not present, that is the cells which are not red, is the place where we can see entry. Hence, the conclusion for this objective is that SARS-CoV-2 membrane fusion is dependent upon the presence of phosphatidylserine in the target membrane. This brings me to my objective 3, which is the final project from a PMRF proposal, and that is the single molecule investigation of spike phospholipid interaction during SARS-CoV-2 fusion to target membrane. Now, in order to understand the single molecule interaction, in order to directly visualize that, I designed a single molecule FRET assay or an SM-FRET assay, where the donor molecule was present in the proteoliposomes and the acceptor was present in the 836 position of the spike. Now, if these two are not fusing, then we'll have a low FRET state. However, in the optimal condition, which I have already explained earlier, then these two membranes will be fusing and we will have a high FRET state. Now, interestingly, we got very interesting data, and for the first time we have shown that at low pH only. When calcium is not present, we are seeing a low Fred state, which is indicative of no fusion taking place. However, uh, when calcium is being introduced in the system, then we are seeing a high Fred state, which is indicative of fusion taking place. This shows that SARS-CoV-2 spike interacts with phosphatidylserine at low pH only and only when calcium is present. On the other hand, if we change the pH to neutral pH, which is pH 7, then whether calcium is present or not present, the SARS-CoV-2 spike cannot interact with phosphatidylserine. Now, all these experiments were carried out under equilibrium state. So we wanted to do a non-steady state measurement of the conformations of the spike. So for that, we used this kind of setup where we float the buffer while imaging itself. And these, the white arrowheads indicate the virus liposome complex. Now, if we look at the data, it is very interesting to see that the SARS-CoV-2 spike is transitioning from a low Fred state to a high Fred state at low pH and calcium. This is for the first time that we have shown this transition in real time for the D614G spike and the Delta spike. Now, another question which arises is that phosphatidylserine should be present on the viral membrane itself, right? So in order to ascertain that, we designed a simple imaging assay where we had SARS-CoV-2 virus-like particles and we used MRF LAC-C2 recombinant proteins which would be binding to the phosphatidylserine present on the SARS-CoV-2 membrane. And here we can see that wherever on the membrane, it is the MRF LAC-C2 is coinciding with the membrane, showing that phosphatidylserine is indeed present on the membrane of the virions as well. Now, the question which arises immediately is that if phosphatidylserine is present on the virions of the membrane itself, then the spike should be able to interact with the phosphatidylserine on its own membrane. In order to answer that, I designed another SM-FRET assay where the donor is present on the membrane of the virion itself and the acceptor is present at the 836 position of the spike, which is the fusion peptide proximal region. Now I saw that at low pH again the spike is not interacting with phosphatidylserine. However, at low pH in presence of calcium, the spike is able to interact with the phosphatidylserine present on its own membrane. Now this will present a difficult situation, right? That if the virus can interact with the pH present on its own membrane, then what if the virus gets inactivated? How will it be able to fuse to the target cell? So to find the answer to that question, I designed an SM-FRET assay just to probe the reversibility of the conformation which is achieved when the spike is interacting with the PS on its own membrane. So here it is the same SM-FRET assay which I have explained earlier. However, what I am doing is that after an incubation with low pH and calcium, I am introducing EDTA. So EDTA will chelate all the calcium present in the system and will be able to find out if this conformation is reversible or irreversible. Now, interestingly, I found that when the virus is interacting with the PS on its own membrane, then after introducing EDTA in the system, the high Fred state is coming back to the low Fred state, showing that it has a reversible conformation. Now, what happens when the PS is present on the target membrane? So I perform the same assay again in presence of EDTA, and I saw that very interestingly that when the virus is interacting with the PS present on a target membrane, then this kind of conformation achieved is irreversible. The high Fred state is not going back to the low Fred state. Now this shows that the post-fusion conformation which is being achieved in the presence of a target membrane is irreversible, and hence the virus is able to fuse to the host cell. On the basis of that, I have presented a model, and this is the first time that any study is reporting how the SARS-CoV-2 spike interacts with phosphatidylserine when it's present on its own surface as well as when it's present on a target membrane surface. So if it's present on its own surface and it's interacting, it is some kind of post-fusion like intermediate state, which we have not been able to characterize yet. However, if the phosphatidylserine is present on the target membrane and when the optimal conditions of low pH and calcium are met, then it has a stable, thermodynamically stable post-fusion state. Physiologically, again, if we go back to the model which I presented earlier, then SARS-CoV-2 entry and fusion is happening via endocytosis when endosome is maturing and the optimal pH and calcium are being reached. Along with that, phosphatidylserine is present on the inner membrane, and hence once the post-fusion conformation is reached, then and only then is the viral RNA being able to be released inside the cell. The final conclusions from my proposal are: first, low pH and calcium trigger SARS-CoV-2 spike-mediated membrane fusion. Interaction with phosphatidylserine in the target membrane is important for SARS-CoV-2 membrane fusion, and SARS-CoV-2 spike fusion peptide interaction with PS in the target membrane triggers membrane fusion. So these are my publications which have stemmed from my work. I have presented my work in international conference, in GRC and national conference. Thank you so much for being patient and listening to my presentation.